Hi, everyone. Today we have as our guest Martha Streng. Martha received a bachelor's degree in neuroscience from Mount Holyoke College in Massachusetts. Then she went to University of Minnesota, where she did a PhD with Tim Ebner. She was instrumental in studying the activity in the Purkinje cells, particularly the climbing fiber inputs to the Purkinje cells during arm movements in macaque monkeys, and made really interesting discoveries about the representation of error, representation of how the climbing fibers encoded events that were in the future, describing the inputs to the Purkinje cells. Then she began a postdoc with Esther Crook Magnuson, who introduced her to the relationship between the cerebellum and the hippocampus. And she's going to talk a little bit about that today, describing the role of the cerebellum in potentially affecting epileptic seizures, linking the DCN and the hippocampus, and some of the discoveries regarding how excitation of the vestigial provides control over temporal lobe seizures. Martha, thank you so much.
Thank you, and thanks so much for the invitation for this really awesome series. I'm excited to talk about some of kind of the recent work from the kind of tail end of my postdoc, looking at interrogating cerebellar network dynamics during both healthy behavior and disease states like epilepsy, as well as some kind of future directions for my own research lab as I'm getting started here at the University of Minnesota. So luckily, most of the time when I start this talk, I have to start it with essentially the pitch of, here's why you should care about the cerebellum. But with this group, I don't have to do that. But obviously, we all know cerebellum is a small volume relative to the number of neurons it has. It's got a very large surface area. It's got all these reciprocal connections. And while it's been predominantly considered within the framework of motor control, it's got ever-emerging roles in cognition, neurological, neurodevelopmental, and psychiatric disorders. So it's very fertile territory for kind of exploring potential roles beyond just traditional motor frameworks. And so, historically, when we look at neurophysiology of the cerebellar cortex, a lot of work has been focused on this kind of particular microzone of, Purkinje cell inputs from parallel fibers and climbing fibers, and their modulation and how they interact with the deep cerebellar nuclei. So this is a lot of work that I was even doing as a PhD student, as Reza mentioned, recording individual Purkinje cells, the complex spike and simple spike activity of those cells, and seeing how they correspond with different behaviors. But as a part of a lot of this work, what really started to become a major interest for me is understanding not only how these Purkinje cell outputs, or these different signals of Purkinje cells are modulated with behavior, but how they all kind of come together at the network level. And there's a couple particular reasons for that. The first is that the cerebellum obviously has this beautifully conserved, relatively conserved cytoarchitecture, but there's a lot of really interesting spatial aspects to those different inputs that modulate both the simple spike and the complex spike firing. So, again, we've got parallel fibers that pass orthogonally through dendritic trees of Purkinje cells, and they have this kind of more transverse type of orientation, whereas the individual Purkinje cells that receive different inputs from inferior olivary neurons, they tend to be clustered in this more kind of parasagittal orientation. So while we focus a lot on the individual signals conveyed by individual Purkinje cells, I think there's a lot of evidence to suggest that the way in which those signals come together must have a very strong spatial component. And so I'm very interested in exploring that. And the second, of course, is that, even though we've got relatively homogeneous cytoarchitecture, there's a lot of evidence to suggest regional differences. So, for example, the expression of different molecular markers can vary, and these different expression of molecular markers can correspond with changes in overarching Purkinje cell dynamics. So the frequency of simple spike and complex spike firing, for example. And then there's also the different lobules of the cerebellum. When we look at different disease states, there's also some potential evidence for regional differences in terms of degeneration of Purkinje cells, and spinocerebellar ataxia can impact more anterior versus posterior lobules in different phases of the disease state and whatnot. So it suggests that, while it's really important to understand kind of individual, single cell firing properties, we also should try to be integrating this into the larger framework of these kind of more network level dynamics to get a fuller understanding of kind of what the cerebellum does and how it does it. So fortunately, I had an opportunity to explore this type of question, looking at these types of spatial scales in awake behaving animals through a collaboration with my PhD mentor, Tim Ebner, and Suhasa Kodandaramaya, who's a professor in the Department of Mechanical Engineering here at the University of Minnesota. So they had previously developed a method for wide field or mesoscale imaging of the neocortex in awake behaving animals through the development of these polymer windows. So essentially they consist of a 3D printed frame that has a clear plastic film called PET that's it's epoxy to that frame. And then we perform a large craniotomy and replace the skull essentially with this 3D window. And so this was done in the neocortex, and we essentially decided to modify this approach to target the dorsal surface of the cerebellar cortex. So it's the same sort of framework, but then we also have a head fixation implant that allows us to image the dorsal surface of the cerebellar cortex chronically and in awake behaving animals. We were excited about this approach to be able to look at kind of network level cerebellar dynamics during behavior. And of course, we wanted to first look at Purkinje cells. That's my and Tim's comfort area. So we took a transgenic approach to expressing GCAMP in Purkinje cells by crossing PCP2 Cree mice with an AI162, a Flox AI162 line, which induces the expression of GCAMP6S in any cell that's expressing Cree recombinase. So in this case, it would be Purkinje cells. And what we were kind of surprised to find with this transgenic approach to GCAMP expression is we were getting expression of our GCAMP that was restricted to Purkinje cells, but it was not expressing in all Purkinje cells. So we got kind of a sparse expression of our GCAMP in these Purkinje cells. And what this allowed for, which was kind of unique, is that we could look at and characterize the modulation of both individual Purkinje cell dendrites as well as the actual somata themselves. So in this zoomed in field of view, you can see the round circles would be Purkinje cell somata, and the thin little strips that come up from each of those somata would be the dendrites of the individual Purkinje cells. So we were not expecting to see this kind of sparse expression, but we decided to leverage it to our advantage because we could then resolve both somatic and dendritic modulation of Purkinje cells. So this is what this looks like in vivo. So this is an awake behaving mouse. So we've got this more mesoscale field of view, and then I've zoomed in on different portions here so you can see that dendritic and somatic modulation. So the dendrites are going to have individual calcium events because those correspond with climbing fiber discharge. Whereas the somatic domains of Purkinje cells, we can see changes in fluorescence, but they're not that more event-based change in fluorescence that we see at the level of the dendrites. And so again, this approach, we can use this chronically in awake behaving animals, and it gives us optical access to lobules 4, 5, 6, and 7 of the midline, as well as regions of the simplex and Crus1. So we can get a pretty large field of view here in awake behaving animals. So we were excited to use this in the context of behavior. And so then the question was, how do we decide to segment a wide field calcium imaging data set like this? So I wanted to take as agnostic of an approach as possible. So what we decided to do was utilize spatial independent component analysis. This is essentially a blind source separation algorithm, and it takes a data set and it segments that data into components that have maximal statistical independence from each other. So essentially it's saying, how does the data want to segment itself based on its functional properties? And the benefit of this is that we don't have to make any a priori assumptions about what the structure of our data is going to look like. So the analysis pipeline is we perform dual wavelength imaging. This is essentially blue and violet LEDs that are interleaved. This allows us to correct for any changes in GCAMP fluorescence that are not due to calcium dynamics. So it's a hemodynamic correction. And so we perform that hemodynamic correction as well as a registration step, which allows for movement correction. We compress the data using singular value decomposition, and then we feed that compressed data into our spatial ICA algorithm. And so essentially you run spatial ICA and it gives you a certain number of independent components. And so what that independent component looks like is a Z-score image where each of the pixels here is color-coded based on the correlation of the dynamics of that pixel relative to the independent component. And so we take those and then we threshold them such that any independent component has to have a z-score of 3 or plus or minus 3 or greater to be considered significantly correlated with that IC. And so this is our thresholded IC. So you can see this is one type that we get when we run a bout of spatial ICA, where we can see these kind of parasagially oriented clumps of Purkinje cells that form these kind of independent components that look like this. But in the same run of ICA, we asked for, for example, 60 independent components, we were seeing a lot of independent components that had this parasagittal type of orientation. But then we also noticed some independent components that had a much more distributed punctate-like distribution throughout the whole image. And when we overlay these with our actual image of the cerebellum, we saw that they overlaid pretty nicely with individual Purkinje cell somata. So we had these parasagially oriented independent components, but we were also seeing these more distributed somatic independent components. And importantly, when we look at the change in fluorescence for those independent components, you can see, so this is an individual Purkinje cell somata that belongs to this particular independent component. We can see changes in fluorescence that have some, calcium event-like looking things, but then they also have this kind of slow time course of increases and decreases in fluorescence respectively. Again, this is data that is hemodynamically corrected, so we don't think that this is due to any calcium independent changes in fluorescence. It's also motion corrected, so we don't believe it's due to any changes in movement. As an important step, we actually take that 405, so that violet signal from that same somatic domain, and we look at the change in fluorescence. We're not seeing anything that looks like the signal that we see here. So again, that also suggests that it's not due to any type of artifact, and that it isn't in fact a calcium dependent change in fluorescence at the level of the Purkinje cell somata. So across the population, this is what some of our different independent components look like. So these are these parasagially oriented independent components, and then these are somatic independent components. And so you can see some pretty striking spatial differences in those. So the parasagittal components are these kind of predominantly consist of one domain that has this parasagittal orientation. Importantly, this parasagittal orientation is very consistent with this beautiful study that was looking at a very large field of view in the cerebellum using glass cover slips that was published in Cell Reports, where they were seeing these kind of more parasagially oriented independent components. This study was imaging only dendritic modulation of Purkinje cells, and you can see ours are a little bit more kind of spatially fragmented. We think this is due to the sparseness of our GCAMP expression, and in fact, when we take our same approach, but we use a viral strategy for expression of GCAMP. So this is a PCP2 Cree mouse that's been retroorbitally injected with a vector for GCAMP6F expression, where we've got expression throughout all Purkinje cells in the cerebellar cortex. You can see they're less fragmented and they look a lot more similar to this, the independent components that this paper sees. So we think that these changes in our dendritic ICs are due to that sparse expression. And then also the fact that we see these somatic ICs suggests that it's because we have this sparse expression and we can see the changes in fluorescence at the level of Purkinje cell somata is why we see these somatic components. So we wanted to look and see, how are these things being modulated during different behaviors? So I'll just show one example from this study. This paper is on bioRxiv, but we looked at kind of how these different independent components were modulated during a cued reaching task. So essentially these mice are head fixed. They're trained to sit in a little 3D printed chair, and there's a water spout in front of them. And they're trained to wait for an auditory cue, and that's denoted by this tone. And then after a fixed interval, there's a water droplet that's delivered to the end of the spout, and the mice make a reach for that actual water droplet, and they can actually physically grab the water droplet and bring it to their mouths and drink the water. So we can look at how the cerebellum is engaged during different aspects of this task. And at the level of just overarching changes in fluorescence, we can see increases in activation throughout the cerebellar cortex that are occurring during the reach, which is not entirely surprising. One particularly notable thing is that the engagement that we see in the cerebellum, this is a unilateral limb movement, but we're seeing very much bilateral engagement of the cerebellum. So this is looking at all of the trials for one recording day in one particular mouse, where we're having changes in fluorescence in both our parasagittal and our somatic domains, where we can see these kind of large increases in fluorescence that emerge shortly after the water droplet is delivered and when the mouse is actually executing the reach. So the touch point here is where the mouse actually physically makes contact with the spout because we've got a capacitive sensor in the spout itself. The other task that we looked at in this kind of framework was we wanted to introduce the concept of a prediction error. And obviously a lot of the motivation for that is because of the cerebellum's role in prediction error processing and things like that. So what we implemented after we collected all of our baseline reaching data is we implemented a catch trial paradigm. And so in this paradigm, each stack of imaging data that we collect consists of 8 auditory tones. And so then in that particular stack, we randomly select one of those auditory tones will have the water omitted. So the mouse is trained to expect water and make a reach after an auditory tone, but in a subset of those auditory tones, we omit the water. And in general, the mice do not reach when what I'm calling the catch trial here is when there's no water that's delivered. So we've got a large difference in behavior as well as a large difference in expectation. And so we were very surprised to see, despite these dramatic differences, that we see very similar patterns of activation at the level of delta F over F or changes in fluorescence when we look at our different somatic and parasagittal domains. So we were very surprised by this finding. And in fact, when we look at breaking this down a little bit further to looking at the actual, when the mice receive water and they reach for it, the catch trial when they don't receive water and they don't reach, but then also cases where the mice receive water and for one reason or another, they fail to reach. They just don't initiate a reach during the task. When we look at across all of our population of animals, we see remarkably similar patterns of activation that are occurring. So the top rows here are for conditions when the mice receive water and they make a reach. The middle is when they receive water and they fail to reach. And then the bottom is when they do not receive water and they do not reach. So you can see when we're looking at z-scores of overall activity levels across our entire population, we see broad bilateral engagement of the cerebellum, both in our parasagittal and our somatic networks of Purkinje cells. So very different behavioral outcomes, remarkably similar patterns of activity. But of course, with a technique like this, with wide field imaging in awake behaving animals, we can look at not only overarching activity levels across the population, but then we can also look at how those activities interact with each other. So what the approach I decided to take with this is looking at essentially spatial correlations. So how correlated are my different independent components to each other during different behaviors as a function of what their relative spatial location is to each other. So essentially what these spatial correlation maps look like is, again, it's the average correlation across all of our different independent components as a function of their relative spatial positioning. And so the benefit of this also is we can look at across our whole population of data. So I can compute these spatial correlation maps across all of the data. This is spatial correlation at rest. And then I can look at how those spatial correlations change during different behavioral conditions. So for example, when that auditory cue is delivered, is there a change in my spatial correlations from rest? And so on. And so if we recall, again, we see large broad bilateral engagement during our reaching paradigm. But if we look at our spatial correlations compared to rest for our parasagittal networks, when we look at the cue period, we're not seeing too strong of a difference. So in this, warmer colors would be increased correlations, cooler colors would be decreased correlations compared to rest. So during the cue period, there's not a whole lot going on in our parasagittal networks. During the reach period, we see kind of a broad decrease in correlated activity throughout our networks, which is kind of interesting because you're actually executing a reach, you're having a large increase in overall activity, but we're essentially having a reduced correlation across that activity. When we look at our somatic networks, we see some really interesting effects that begin during that actual cue period, in contrast to our parasagittal network. So during the cue period, we're seeing an increase in correlations, but in particular, it appears to be along that transverse axis. So Purkinje cells that are along the same transverse axis tend to be increasing their correlations more so than those that are off of the same transverse axis. And we see actually the reverse during the reach itself. So again, we see strong decreased correlations, but appears to be more for Purkinje cells that do not share the same sort of transverse region. And this is what it looks like quantified when we take this kind of middle third versus the distal thirds, where we see a large increase in the more proximal Purkinje cells during the cue period, and then a decrease in the more distal Purkinje cells during the reach period. And I think this is a particular interesting finding when we consider the cytoarchitecture of the parallel fiber inputs that are likely, contributing to changes in Purkinje cell somatic modulation, that the correlation dynamics of those Purkinje cell populations appear to obey that cytoarchitecture of those parallel fiber inputs. So we're seeing some potential functional evidence for the structure of those inputs at the level of the somata. And we don't see it in our parasagittal networks. It's specifically in our somatic networks, which is kind of cool. So how about during our catch trial period? So again, this is where we see similar activation in terms of our standard reach versus our catch trial condition where they do not receive water. And we were particularly surprised by our correlation dynamics when we look at comparing our parasagittal versus our somatic network. So our somatic networks are showing dramatic increases in correlated activity when we compare the correlations during the catch trial versus the standard drop trial. So when the mice expect water and they don't receive it, there's a huge increase in how correlated those somatic networks are. And again, just to remind you, this is despite very, very similar patterns of activation. So this technique is allowing us to capture not only how different areas of the cerebellum are being activated, but it appears as though, especially in the case of this paradigm, that it's the interactions between those networks that are showing the most dramatic modulation compared to standard conditions, compared to just overall activity. And we wouldn't have been able to see that if we hadn't had this large field of view that we can use in awake behaving animals. So a final question I had in the case of this study was, okay, if we see these large changes in correlation dynamics, what is the time course of those correlation dynamics looking like? And in particular, I was also interested in, can we see behavioral outcomes within those changes in correlations? And so I actually returned back to the situation in which the mice receive water and choose to drink versus receive water and choose not to make that reach and drink. And I looked at, with using a sliding window, the temporal progression of changes in correlations, comparing those two conditions. So again, in this particular paradigm, the mice are always expecting water and they're always receiving water. And the blue line denotes the correlations across the somatic networks when the mice decide to make a reach, and then the red is when the mice fail to reach. And it's aligned to the different time points of the task. So here's the first time point when the auditory cue is delivered. And interestingly, we can even see significant differences in our network correlations in the time just before the auditory cue. So when mice ultimately fail to make a reach, they have significant decreased somatic network correlations before the timing of the auditory cue. And then when the cue is delivered, we see an interesting change that occurs even before the water droplet is actually delivered. So if you recall from the spatial correlations, we saw an increase in correlations along that parallel fiber axis, the putative parallel fiber axis. And we see that increase reflected here, and you can see the temporal progression of it. The mice that fail to reach, or the trials in which they fail to reach, there's also an increase, but it's significantly reduced compared to the successful reach conditions. Now, when we look at the actual reaching window, so this is the time from when the water was delivered to the maximum time allowed for the mice to actually make that reach, we see a very cool temporal progression of those correlations. So again, there's a decrease in correlations when the mice make a reach. It reaches a local minimum within that maximum reaching window. And when we look at the fail to reach condition, there's also an initial decrease in correlations, but then there's this inflection point where it returns back to the baseline correlation. So it's almost like we can see, the point at which the mouse, for one reason or another, decides not to make a reach or fails not to make a reach. Obviously, I can't ask the mouse at what point it made that decision or, whatever, but it certainly looks like we can see some kind of neurophysiological correlate of that success to reach or failure to reach within our correlation dynamics. And even when we look at the, immediately after the post-reach time, we can still see a significant difference in those correlations. But you can see from this sliding window that there's very dynamic properties of how correlated those somatic networks are. And then I'll just show you during the catch trial condition, again, we see these strong increases in correlations, that we saw during the spatial correlation analysis. But one interesting thing to note from this, so the blue line is when the mice receive water during this paradigm, and then they reach for it. Again, just to remind you, all of this data was collected before this data. So instead of seeing two blue lines that look similar across conditions, yes, we see a decrease in correlation during the actual reaching window, which makes sense, but we've lost our increase in correlation during that auditory cue period. So it's almost like we're seeing evidence of that reduced certainty about a reward that's going to be delivered after the auditory cue. So now that the mice no longer know if every auditory cue is going to result in a water droplet, we no longer see that increase in correlation during that auditory cue period. So again, this is potential correlates of actual behavioral outcomes that we can see in our correlation dynamics across our somatic networks. So just to summarize this part of the talk, I showed you kind of our analysis of this wide field imaging data that we performed chronically in awake behaving mice, where we see these distinct functional networks of Purkinje cells when we look at sparse GCAMP expression. So we've got parasagially oriented Purkinje cell networks, and we've got these more somatic, more distributed Purkinje cell networks. And we use the dynamics of these different networks to show that different behaviors result in potentially divergent impacts on the network activation versus correlation. So, despite the fact that we can see very similar changes in overall activity, it looks like the interactions across those activity modalities are conveying a lot of information about behavioral outcomes. And of course, I also more recently I showed you that we can see kind of potential behavioral outcomes reflected, in those actual correlation dynamics. And some of my ongoing and future work with respect to these questions in particular is, of course, what are these correlation patterns reflective of? So in particular, in the no reach condition, the reduced correlations that we see in this inflection point that we see, what is that reflective of? Is it reflective of, failure to attend appropriately? Is it reflective of change in motivation, related to things like satiety across a trial, other things like that. So I'm excited to kind of explore some of those future directions within my own research lab. So the next study that I'll just tell you, and this is a lot more recent work that we've been doing, is looking at leveraging this technique to explore how the cerebellum is modulated during hippocampal seizures in temporal lobe epilepsy. So that might seem like a total left turn. I was in, very much motor control domain looking at healthy behavior, and now all of a sudden I'm talking about epilepsy and seizures. It probably feels like a left turn because the cerebellum is not traditionally associated with epilepsy or seizures. It's actually been historically considered, there's been this persistent view that it's one area of the brain that doesn't seize in general, but there's been a lot of evidence to show that not only is the cerebellum involved in seizure networks, but it's a potentially critical node. So this is some of my work that I was doing earlier in my postdoc with Esther Kirk-Magnuson, where we were exploring optogenetic manipulation of cerebellar neurons to stop hippocampal seizures in a mouse model of temporal lobe epilepsy. And we showed that excitation of these neurons in the fastigial nucleus could profoundly attenuate hippocampal seizures. As Reza mentioned, we showed a kind of interesting directional selection effect where inhibition had no effect, excitation is required. So there's a nuanced relationship between the cerebellum and hippocampal seizures in particular, but it appears as though the cerebellum can powerfully influence those seizures. And as we know, since the cerebellum is hallmark is these reciprocal loops, it might be unsurprising then to know that if the cerebellum can influence seizures, there's a lot of evidence to suggest that the seizures themselves can influence the cerebellum. And in fact, the manner in which these seizures influence the cerebellum has particular implications for patients. So in patients who have epilepsy, there is a negative outcome that is particularly devastating that can occur. That's called sudden unexplained death in epilepsy. And this was a recent study that looked at kind of changes in brain volume of different regions of the brain that were associated with either high SUDEP risk or ultimately in patients who went on to have SUDEP events. And the only two areas of the brain that showed significant volume reduction in cases of high SUDEP risk or actual SUDEP were the hippocampus and the cerebellum. So it suggests not only that, again, that these seizures can influence the cerebellum, but if cerebellar alterations occur in epilepsy, it can potentially be contributing to negative outcomes in patients. So it's very important to then fully characterize how the cerebellum is impacted by chronic epilepsy as well as spontaneous seizures. There's been very limited work on this. Some early, some early studies looking at cerebellar engagement in different animal models. This is a more recent example from Esther's postdoc work, where they did juxtacellular recordings of Purkinje cells in animals that had chronic hippocampal seizures. And they saw that when the seizure emerged, we could see, phase locked, essentially Purkinje cell firing to the different spikes within an actual hippocampal seizure. But again, this has been very limited. It's often restricted to specific populations, and it's restricted to specific models of epilepsy. And part of that is just because the cerebellum has been historically very ignored in the context of temporal lobe epilepsy. So we had this great opportunity to, of course, leverage our technique, which since we can do this chronically in awake behaving animals, we can look at these more mesoscale level changes in cerebellar activity during spontaneous seizures. So this is what this model looks like. Essentially, mice are injected once unilaterally with intrahippocampal chinic acid. This produces a large status epilepticus event, and the mice then recover from this event, and over a period of weeks, they develop chronic spontaneous electrographic hippocampal seizures. So this is an example video of this. These seizures are focal, meaning that there's not a motor component. So we classically think of a seizure as having this tonic-clonic sort of thing. There's no behavioral component to the seizure, and so you'll be able to see this in a minute. So this is the mouse running on a freely moving disc treadmill. This is a cerebellar signal that we're simultaneously getting. And then this is a hippocampal seizure that emerges. So you can see the animals continuing to run. It'll pause for a little bit at a certain point in time, and then it'll start running again. So these are often referred to as subclinical, focal, electrographic seizures. It's a hallmark of this model. The majority of the seizure events in these animals are electrographic only. And so the other thing I want to note is that we didn't give these animals anything to induce these seizures. They get the initial intrahippocampal chinic acid injection, but then the seizures just happen when they want to happen. So we're not giving them anything to make them have them more frequently. We are just recording from them over and over and over and over again, and hoping to capture a seizure. And so for that reason, I want to highlight that this study would not have been possible without several people who contribute a lot of time and energy to collecting this data. So Ben Kotke, Lucas Zucker, Emma Wasserman, and Lin Wang were very instrumental in tirelessly collecting day after day after day of data in these animals, trying to capture some of these seizure events. So we did capture some of the seizure events, and we wanted to look particularly at what's happening in the cerebellum at the onset and the offset of these hippocampal seizures. And so what we saw was something pretty remarkable, which is that we're seeing changes in cerebellar activity that are beginning just prior to the onset of a hippocampal seizure. So this is from an example mouse example day. It's aligned to that onset. You can see this is the average LFP across all of the different recordings. An example event is pulled out in black here, where the seizure begins, but we're seeing widespread depression throughout the cerebellar window beginning just prior to the onset of that event. And so I'll just stress that this model, the hippocampus is the seizure focus. So I'm not saying that the seizure is beginning in the cerebellum per se, because the seizure should be beginning in the hippocampus. That's the site of the insult of the chinic acid injection. But what we're seeing is more potential hints, I think, of seizure susceptibility, whereby the cerebellum might be a really key player. So if you have widespread depression of the cerebellum, it might be signaling the entrance of the brain into a more seizure-prone state. And so if you've got broad bilateral decrease in the activity of the cerebellum, it might be, the downstream effect might be that the hippocampus is more liable to have a hypersynchronous event. And when we look at across the whole window, we organize our changes in fluorescence by the anterior-posterior positioning. We see a pretty cool gradient whereby the more posterior regions of the cerebellum, so again, in our window, that's going to be more lobule 7, the more posterior regions of the cerebellum are showing the strongest decrease in activity prior to the onset of that actual hippocampal seizure. So instead of seeing kind of broad bilateral changes, we're seeing the strongest in our more posterior regions. When we look at the offset of a hippocampal seizure, so the organic termination of a seizure event, what's really neat is we see the opposite effect when we look at the activity of the cerebellum. So around the timing of event offset, we see an increase in activity throughout the window. The strongest increases are observed in those more posterior regions. And again, even though this increase is peaking after the time that the seizure stops, this is comparing a GCAMP6S signal with an electrographic signal. So it's very likely that this peak is happening around the time of the event offset, and it's certainly beginning just prior to the offset of that event. So it suggests that organic activity levels of the cerebellum might be directly contributing to the termination of a seizure. And this kind of, the reason why I use that language is it kind of mirrors the optionetic work very nicely, whereas before we're experimentally altering the activity of the cerebellum. Here we're seeing, just looking at a spontaneous seizure that terminates, we're seeing those kind of similar types of dynamics that are occurring, in the baseline state when we're not performing any particular manipulation. And in fact, when we look across all of our population of animals, so we had over 600 events from 6 different mice, you can see that depression just prior to the onset and the rebound that begins prior to the offset of those hippocampal electrographic seizures across the whole cerebellar window, looking across the whole population of animals. So we're seeing this very robust kind of reciprocal change in dynamics that are associated with the onset and the offset of spontaneous hippocampal seizures. So again, because everybody was, contributing so much effort and recording from mice over and over and over again, we were able to capture something that at the onset of this study, I never really thought would be possible, which is that we were able to capture spontaneous behavioral seizures. So these events occur very rarely. It's essentially an electrographic seizure that generalizes, and it does become a tonic-clonic event. Individual mice can have these essentially once a week. And so we got very, very lucky, and after many persistent efforts, to be able to capture some of these behavioral seizures. So these do have a behavioral component. They're a hypersynchronous event where the animal has tonic-clonic seizure. I'm obviously not going to show you the behavior of that. I'm going to show you what's going on in the cerebellum during that. Basically, this hypersynchronous activity of the hippocampal LFP is when the behavioral seizure emerges. So this video is a minute and a half, but I think it's worth it because this is, to our knowledge, the first imaging data during a truly spontaneous behavioral seizure that's been collected. And lucky for the epilepsy field, it's been collected in the cerebellum and not the neocortex, so they're going to have to give the cerebellum the appreciation it deserves. But what we see is a really interesting and very different pattern of activation throughout the window compared to our electrographic events. So you'll see prior to the onset of that behavioral seizure, it begins right now, there's a broad increase in activation of the cerebellum. And this is prior to any motor manifestation of the seizure itself, and it's prior to the hypersynchronous, this is the actual behavioral seizure happening, any hypersynchronous activity in the hippocampus, we're seeing this broad increase. And then, particularly in the anterior regions, they're going to become very reduced in activity, and there's this cool posterior wave that kind of emerges. There's going to be 2 bouts of it, and it's going to peak right at the end of the seizure, and then the whole window is going to go dark during the postictal stage. So it's peaking here, and then the whole window goes dark. So very, very dramatically different dynamics, but importantly, those dynamics begin several seconds prior to the onset of the actual hippocampal seizure. So this is what this looks like for the seizure when we look across all of our different window, where you can see this increase in overall activation that occurs prior to the onset of that hippocampal or behavioral seizure event. You can see the depression in the more anterior lobules and then the widespread depression during the postictal period. This is some example traces from some different example somatic domains. So you can see it's occurring both in the parasagittal and somatic regions, where it's the most pronounced in the posterior regions, and the anterior regions start to become more depressed even during the seizure when it's ongoing. And I think that's really interesting because it's the more anterior regions, lobules 4, 5, 6, that are going to be considered more motor based. And while the animal is still convulsing, essentially, we see a silencing of those regions. So this is those same heat maps for this particular example seizure. And again, never thought we'd ever have 1 behavioral seizure, much less 11 behavioral seizures, so this is the heat maps for those, but you can see very different impacts of the seizures and what's going on in the cerebellum even prior to the onset. So dramatic increases in activation that are beginning several seconds prior to the onset of that behavioral seizure. So the last data I'll show you is looking at interictal discharges. So basically in this model, we've got electrographic seizure events that are denoted here by this orange bar. But then between seizures, you will see epileptiform discharges or interictal spikes. And so while these seizures can produce changes in activity within the hippocampus and within the contralateral hippocampus, interictal spikes even aren't necessarily seen in the contralateral hippocampus. So it's not necessarily expected that we'd see anything even further away in the cerebellum, but we in fact do. And this was probably the most surprising aspect of this study to me, which is that even intraepileptiform discharges are associated with changes in cerebellar activity. And again, there's this anterior-posterior gradient where the more posterior regions of the cerebellum are more activated by those interictal spikes. And also when we look across the population, we also see changes that begin prior to the actual interepileptiform discharge. So there's a depression that begins prior to that event, and then a rebound that occurs immediately after. So this was really, really, pretty remarkable that we can see stuff like this in the cerebellum, and in fact that the cerebellum might be leading that actual interepileptiform discharge. So it might be directly contributing to whether or not the hippocampus is going to have an interictal spike or not. So we can track a bunch of these over time. And so what I wanted to do was compare the changes in activity that we see relative to interictal spikes. And if we could see differences that might emerge, whether the animal is going to have a behavioral seizure or not. Because, again, one major focus of the epilepsy field is what is going to contribute to seizure susceptibility? What's going to make the animal more likely to have a seizure? And when we look at these kind of rebounds after an interictal spike, we see an interesting gradient that emerges as we get closer and closer to a behavioral seizure. So 30 minutes out from a behavioral seizure, we see the strong rebound that we see in our standard data. But as we get closer and closer, so as these lines get warmer, we lose our rebound. And then we had one animal that we collected data with from over a period of 3 months. So again, this is, benefit of this technique is we can record from this very long period of time. And we look at that animal that actually went on to have a putative SUDEP event, which can happen in this model. We see something very similar. So this is 90 days out from the SUDEP event. We see this beautiful rebound after an interictal spike. And as we get closer and closer and closer to that SUDEP event, so this is within 10 days prior to the SUDEP event, we've lost most of our rebound, and now we predominantly see depression throughout the cerebellar window. So we can potentially be seeing biomarkers of SUDEP susceptibility when we look at our cerebellar activity relative to these interepileptiform discharges. So I think there's some potentially really valuable information here when it comes to those issues. So to summarize what I've shown you, the cerebellum is highly engaged during different hippocampal epileptiform activities. Interestingly, that modulation appears to begin prior to the onset and the offset of the events, suggesting that it might be a very active role in the seizure network from the seizure susceptibility perspective, and that it might contain value information about whether or not a seizure is going to generalize and become a behavioral seizure, or how susceptible the animal is to having a SUDEP event. So as I'm getting my lab started, I'll just mention a couple future directions that we're working on. So we're working on right now looking at cerebellar structural changes in our particular model of temporal lobe epilepsy. So this has been done in a couple different rodent models, but not in temporal lobe epilepsy models. And we're starting to see some pretty cool evidence that there is Purkinje cell degeneration happening in chronically epileptic mice, but that's particularly pronounced in these posterior lobules. So this is work that's being done by Ben Kotke, who actually came with me when I started my lab. And then also Alethria Voyagolozu, who's been getting started as a technician more recently. So they've been working really hard on this histology project, and we're looking forward to seeing where this goes. I'm also looking at exploring the cerebellum beyond temporal lobe seizures. So we've got a cortical model of epilepsy where we've got cortical epileptiform events, and I'm going to be looking at how the cerebellum is modulated during these events, and then also returning to some of my optogenetic work to look at whether we can target vestigial outputs to terminate those events. Finally, I want to get back to these kind of exploring these more spatial organizations more in the context of behavior. So using some intersectional approaches to further dissect these networks, not just looking at Purkinje cells as a whole, but potentially Purkinje cells based on the specific inputs and outputs that they receive. We've got some cool intersectional tools to be able to target our opsin or GCAMP expression, particularly in these populations. I'd like to explore these kind of different molecular markers and how they correspond to our different Purkinje cell networks. And then, of course, I'd love to get back to being able to look at spikes because that's my happy place. GCAMP is never going to give us simple spike firing. It's just too high frequency. And so I've been really thrilled to have the opportunity to collaborate with two relatively new folks here at the university, Madhu and Ganesh, who developed genetically encoded voltage indicators. So they're pretty much, developing these top of the line tools. And we've been able to express those indicators in Purkinje cells. And when we drive our imaging system fast enough, we think, obtain fluorescent simple spikes and complex spikes. So stay tuned for some of that work in the future. And then also, Suhasa is incredible at technology development and developing new modalities for imaging. And so I'm working with Suhasa and Tim and a PhD student of Suhasa's, Ibrahim, who's developing a method for head-mounted mini camera arrays to be able to do this wide field imaging in freely moving animals. Because, as we discussed at the beginning, I think especially in the consideration of the cerebellum, having freely moving behavior is going to be able to provide a lot of really valuable insights into cerebellar physiology and function. So using all of these tools to kind of explore healthy contributions to healthy behavior as well as different disease states are kind of the overarching research directions of my lab. So with that, I'll just mention, of course, my previous mentors, Esther and Tim, who, a lot of this work that I presented was, my final postdoc work working between their labs. Ben, Lynn, Lucas, and Emma were instrumental in the epilepsy imaging study of the data that I showed you. That we're hoping to have that on bioRxiv in the next couple weeks, so stay tuned for that full study. And then, of course, my lab is just getting going, but we're a lab of 3 with Eleftheria and Ben. I'm really looking forward to growing the lab as we move forward. So if any of this is a potential interest to you, I really look forward to getting in touch. So please get in touch if you have any questions or interests. University of Minnesota is a really awesome environment. I probably demonstrate that by the fact that I've stuck around for so long. So I've been happy to get the opportunity to utilize all of these really different fabulous resources. And then finally, these are my funding sources. Some of these are Esther, some of these are mine. Yeah, so with that, thank you all for your attention. And in the time we have left, I'd be happy to answer any questions or have any future discussion. So thank you.
Thank you, Martha. And if you have any questions, please just raise your hand and we can ask Martha about it. Martha, can you teach us a little bit about the relationship between the GCAM signal and the dendrites and how it differs from the soma in terms of what you think, if you were recording electrophysiologically, what are the 2 signals you might see?
Yeah, so I mean, the dendritic, obviously, there's been a lot of work using 2-photon imaging and everything like that, we can feel pretty confident that the dendritic modulation is being driven largely by the climbing fiber inputs. And given that our parasagially oriented domains are similar to those that have been observed, imaging just dendritic activation, we think that the parasagittal domains are largely reporting dendritic modulation. And when we look at the actual delta F over F of those domains, we see a lot more of those kind of events, those very high amplitude events that might be more indicative of kind of population climbing fiber events. The somatic domains, obviously, the simple spikes are initiated at the axon hillock. If you record electrophysiology, you get closer to, as you get closer and closer to the Purkinje cell somata, I remember a lot from my graduate work. You drive through the dendritic layer and you're recording just complex spikes. You hit the sweet spot and you got complex spikes and simple spikes. And if you go too deep, then you just start to see simple spikes. So I would think that the somatic modulation is largely reporting, changes in simple spike activity. That being said, there's definitely going to be some, dendritic or complex spike type of information there. So it's a little bit more mixed, which is, I think, why it's really interesting that these spatial independent component analysis, it's agnostic to all of those things, but it's still pulling out somatic networks versus parasagittal networks. So I suspect that somatic modulation contains enough unique or distinct information from the dendrites that it can be pulled out by something like spatial ICA. Of course, we can't get at individual simple spikes, but I think, and I know there's some literature that's done somatic recordings with GCAMP, and it's shown that they do simultaneous electrophysiology, and they can see changes in simple spike firing that appear to mirror overarching changes in fluorescence. So that's what we suspect, but it's certainly a correlational hypothesis rather than a causal one at this point anyway. So
Sounds good. Nikki, you have your hand up.
Yeah, really amazing talk, Martha. I have a question, kind of trying to wrap my mind around how your spontaneous Purkinje cell activity relates to those manipulations that you did to stop seizures. So the recordings here are in Purkinje cells, right?
yes.
And you previously predominantly manipulated nuclear neurons in order to stop. Do you see that there's indeed between this spontaneous activity and your therapeutic intervention that there's an inverse correlation, or do you see that the Purkinje cells and the nuclear neurons kind of seem to be working in a similar direction? I'm just trying to understand. The reason I'm asking is I'm trying to understand how the changes in Purkinje cells may ultimately change the network activity that would make a brain more or less susceptible for a seizure. Sorry, this is a long question, but
no, yeah, no, it's a really good question, and it's something I obviously have been thinking about a lot. So my work was done in the deep cerebellar nuclei, but it was actually kind of a follow-up from Esther's postdoc work where she was targeting Purkinje cells. So she targeted Purkinje cells in either lobule 4 or 5 of the vermis or the simplex. And the really interesting divergence there is she used optionetic excitation of Purkinje cells and optionetic inhibition of Purkinje cells, and both excitation and inhibition were effective at stopping seizures. So it appeared as though just disrupting the activity pattern of Purkinje cells in some way was sufficient. And that's why my first step with that study, looking at the vestigial, I started with inhibition because I thought, excitation of Purkinje cells equals inhibition of nuclear neurons. And it, of course, turned out to be a lot more nuanced than that, which is that we needed a net increase in vestigial output to stop those actual hippocampal seizures. So the difference between Esther's work and this is that obviously we're seeing this over a larger field of view, whereas she was just targeting a particular lobule. So it's possible that, having more spatially restricted interventions means that both excitation and inhibition can work. But, it is really interesting to think about, especially from the perspective of the nuclei, that we needed an increase in output to get seizures to stop. How does that correspond with these broader network level changes that we're seeing, across the cerebellar cortex during the organic onset and offset of seizures? And so I think, the integration across Purkinje cells to nuclei is obviously, it's a fascinating thing to think about, both from healthy behavior, but also from these disease states. So, the probably unsatisfying answer to the question is like, I'm trying to wrap my head around it too. But,
can I ask another follow-up question?
sure.
So in the spontaneous motor behavior, you see that actually some of the most powerful information is not so much in the activity, but in the correlation between the different zones. Have you looked at something similar like that in the epilepsy work?
Yeah. So if I just didn't have the time to mention it. So this is zooming ahead from all my slides here. Let's see. There we go. So I did look at these changes and correlations, and we're seeing some really fascinating network level dynamics. In particular, in the preictal period, so the time before, the 5 seconds before the mouse has either an electrographic or behavioral seizure, the changes in activity that we're seeing are mostly manifest as a decrease in correlations. So the network is becoming highly essentially desynchronized prior to the onset of a seizure, which is like a super, for me, was not a straightforward finding because I think of seizures as hypersynchronous. But again, I think it points to a somewhat, if the activity of the cerebellum somehow is becoming dysregulated in some way, it can be directly influencing the seizure network and how likely it is to have a seizure. The other interesting thing is the end of a seizure is associated with a pretty large increase in correlated activity, in particular for behavioral seizures. And that also gets back to this optionetic work concept of if you stimulate optionetically, you're going to introduce a synchronous increase in activation. And so we appear to be seeing that during the actual organic termination of a seizure itself as well.
Thank you so much. We have Olivia, I think you had your hand up. Did you want to ask a question?
I did, but okay. I was wondering if your work is giving hope to people who have psychogenic non-epileptic seizures.
Yeah, it's a really good question. I think, and that's why part of it is I'd like to get into different models of epilepsy as well, because I think there's a lot of work from both Esther's lab, this work, Laura Rondy-Riggs' lab, that the cerebellum and hippocampus have a potentially very unique and privileged relationship. What I'd like to know is, is the cerebellum potentially, you know, a node that can influence just seizure networks as a whole? So like, can it be a regulator of ictal activity in general, or does it have to be hippocampal ictal activity? And so that's definitely going to be some future directions that we explore within my own lab, is like moving beyond temporal lobe epilepsy models, you know, cortical epilepsy models. There's genetic epilepsy models, you know, especially those like Dravet syndrome that have a much higher incidence of sudden unexplained death in epilepsy. That's definitely going to be like an active research area that my lab's going to be exploring.
Cool.
Yeah.
Thank you.
Martha, what do you think of, I was thinking that it's really surprising that your beautiful results on how increased activity in the vestigial causes the seizure to end. I wonder if you can teach us a little bit about why that might make sense. And also, what do you think of scenarios where the output of the cerebellum is being disrupted, say, some kind of deep brain stimulation, where you are effectively cutting this output to the rest of the brain? And maybe you can tell us a little bit about how that may or may not be beneficial.
Yeah, I mean, I think I was talking with Aisha Thanwala about this. She gave a seminar within our cerebellar group, and she does some, you know, vestigial stimulation and you know, looking at different output channels of the vestigial, but during healthy behavior like reaching. And we were having a discussion about how we think about, especially in the context of the cerebellum as a predictor, how do we think about the idea of applying an external stimulation to those neurons versus some type of more physiological organic change in activity that is occurring during behavior. And I think, you know, sticking an optogenetic probe in and stimulating the vestigial nucleus is going to have a much more dramatic change in downstream activity than just an organic physiological increase in vestigial neuron activity, because essentially you're perturbing the system that's meant to make predictions, you know. And so I think an unexpected stimulus to the vestigial could produce a very dramatic change. And Anjali Neitz in Tim's lab was doing some preliminary work at looking at neocortical imaging during vestigial stimulation, and stimulating the vestigial with optogenetics produces a dramatic increase in activation, essentially, of the entire neocortex. So I think it can really, that from the seizure perspective, I think that can be very powerful at perturbing, you know, a hypersynchronous event, that it could produce this widespread change in the network. But I think especially with the epilepsy study that I showed here with the imaging, there could be an interesting difference in organic changes in activation versus externally applied changes in activation. And so like one of the things I'd really like to do is, you know, rather than doing a closed loop stimulation approach, could we overall augment excitability of Purkinje cells or decrease excitability of Purkinje cells using something like DREADS, for example? So if I just kind of not stimulated a specific time lock point, but if I change the overarching excitability, could I decrease or increase the susceptibility to have seizures in general? So getting beyond some kind of deep brain stimulation, but more kind of global change. But I do think the cerebellum is particularly tricky, at least for me, to think about how we interpret results of stimulation versus recording data. Like, I think it, you know, I think that's the case everywhere, but especially when I think of the cerebellum as a predictor, it's always something that's on my mind. But
Wonderful. Thank you so much, everyone. Any other questions for Martha? All right. Good to see you.
good to see you. Thank you. Yes, thank you so much for the invitation.
A pleasure. Thank you. Have a good day, everyone.